Hey there, Bio 312 students. What I'm going to do in this video is show you how to take your fish COX-1 DNA sequence and compare it to a large database to determine what species of fish you ended up with. If all goes well, it should match closely to what was on the label. We're going to be using a website that is a branch of the NIH called the National Center for Biotechnology Information, or NCBI. NCBI is where the database called GenBank is housed. GenBank is a huge database of DNA, RNA, and protein sequences that is available to the public for uses such as ours. All published research that includes these kinds of sequences are required to be uploaded to the GenBank, so this database is growing on a daily basis. To accomplish our task, we'll be getting the necessary files from Blackboard, so you'll need to download both the forward and reverse sequences. These are the .seq files. Your forward and reverse fluorograms. These are the PDF files. And then we will get the reverse complement of the reverse sequence. And then we'll clean up both sequences by removing any ends at both ends. And then we're going to align these two sequences, which will allow us to get one clean consensus sequence to do our BLAST with. So what I've done here is I've downloaded both sequences, and I've chosen tilapia to work with. And I've got both the forward and reverse sequence. And the first thing that I'm going to do is change the font to Courier New and a 10-point, similar to what we did with our mtDNA. And I'm also going to change the layout of the page here, give wide open margins. And you're going to notice right away that there are a good number of ends in both the beginning and the end of these sequences. But before I clean those up, what I want to do is I want to take the reverse sequence, the sequence that was sequenced with a reverse primer. I'm going to copy and paste that. And I'm going to go to the handout, which you should have already downloaded. And I'm going to get the reverse complement of that. So I'll go to the website and plug that in. And let's grab this. And we'll paste this in. And you want to make sure, now here's the common problem, you want to make sure that you actually grabbed the reverse complement and you just didn't repaste what you had copied before, and that's exactly what happened here. And this does happen, so let's get rid of that. And we'll go back and make sure we grab this. Alright, now that is the reverse complement. So I'm just going to grab this heading. I'm going to title it Reverse Complement. Alright. And the reason why we wanted to get the reverse complement is that we're going to align these two sequences, the forward and the reverse sequence, and to be able to compare them, the reverse sequence actually has to be put into the reverse complement so that we're reading them in the same direction. So we're going to read them both in the 5' to 3' direction. So let's go to the forward first, and we'll go to this second or copy of the forward sequence. I'm going to take out all of the ends all the way to the GAA beginning part there. And let's get it all lined up. And make sure that you do not delete any bases when you're doing this. It's really easy to delete by accident. Okay, don't worry about any ends that might be in the middle of the sequence. Then we're going to go down here to the reverse complement and do the same thing. Oh, first actually, what I want to do is take out the ends at the end here as well. Okay, that's good enough. So let's go down to the reverse complement and we'll take the ends out of the beginning. And I'll actually go up to the GGC here. And then take the ends out of the end. All right, great. So we are ready now to align the two sequences, and so we want to go back to the handout, and we want to go to the NCBI web page, which I've already got open here. And this is the web page that we're going to use to do the blast. So this is the icon that we click on to do the blast, and if you scroll down, you want to click on the global align to do the alignment. So you have two boxes, and you're going to put one sequence in each box. What I would suggest is that in the first box you put the forward sequence, in the second box you put the reverse, so that when it spits out your alignment you can keep track. The forward is going to be the first one and the reverse will be the second one in the alignment. So let's go ahead and grab both sequences without the header. So we'll go down here and grab the cleaned up forward. Put that in the first box. You get the cleaned up reverse complement and put that in the second box. And now we'll hit align. And it won't take very long to do the alignment. And the first thing you want to do is take a look at this graph, and you can see a really nice clean line here. If you've got a lot of breaks in this line, it means that you don't have good alignment, and it's possible that you accidentally made a mistake by maybe uploading the reverse sequence in the reverse order rather than the reverse complement. So make sure you double check that you have the correct one. And here is the alignment. So let's just grab the whole thing, and I would suggest putting it into a new Word document. So here is our alignment, and you're going to notice right off the bat that the alignment, the two sequences are not in 100% agreement, and that's perfectly fine. You want to go through and take a look at both sequences and decide which one you want to use. And that's going to be based on a couple of things: which one's longer and which one has cleaner sequence. And so as I go through and look at the bases and look at the sequence here, I'm going to choose the one that's called subject, which is my reverse sequence. The query is the forward, the subject is reverse. I'm going to choose the subject because it just looks like it's a cleaner sequence. And so what I'll go through is I'll go through and highlight each line of that sequence. And so I suggest you make the same decision, the same choice, and go ahead and highlight yours. I'll see you when you get done with that. All right, I'm back. So I finished highlighting the reverse primer sequence in my alignment, and I also opened up a new window that I can compare side to side the reverse primer sequence, the whole sequence together like this on its own. And so what you want to do is you want to look through the sequence here and decide if there are any ends in the middle that you may want to go and check in your alignment to see if the opposite sequence, the other sequence, gives you a base there. So I don't see any ends here, and what you're doing is you're building a consensus sequence. So you're building your best, strongest, longest, and cleanest sequence to do the blast. The next thing you want to do is look quickly through your alignment and see if there are any bases that do not agree with each other, and I do have one right down here. So you'll notice that in the query sequence, which is the forward primer sequence, we have an A, whereas in the subject sequence, which is the reverse primer sequence, I've got a G. So what I want to do with this is go back to the fluorogram and make sure that that is really a G in your consensus sequence. So this is where you're going to pull out the fluorogram and double check. So that's two things you want to look for. If there are any ends in your sequence that you've chosen, don't worry about the other sequence because you're working here now with, I'm working with the reverse primer sequence. And so I'm not worried about the ends that are in the forward because I'm not going to use that sequence. I'm only concerned about any ends that are in this sequence. I want to go and look at the alignment and see if I can get a base reading off of the other sequence that's there, the other strand, or you want to go to the fluorogram. And especially you go to the fluorogram if you find that there is a disagreement in the bases between the two strands, and you clear those up. Okay, so I'm going to go ahead and do that for my own, and why don't you take the time to do that, and I'll see you back here when you're ready. All right, so I've gone through and I've cleared up any issues with my bases, and I now have my consensus sequence. I'm going to grab that, and I'm going to grab that header as well. And we're going to go to the NCBI website. We'll click on Nucleotide Blast. And we're going to paste our sequence in there, including the header. We'll come down here, and under organism, this will probably be blank. If you start typing bony fishes, it'll come up. So you choose that and you hit blast. And this is going to take a few seconds. So I'll see you when mine gets done, and we'll proceed from there. So here is the blast, and there are a couple of things that I want to point out to you. First of all, on the left-hand side here, in the upper left-hand side, the title of our blast is the header that we included in our sequence. And if we take a look, it's telling us it's a nucleic acid, which we already know, and it's 643 bases long. And so we are comparing, this program is comparing our DNA sequence to a whole database of sequences that have been uploaded in the group bony fishes. Okay, so we've narrowed down our database, which is a good thing to do. And we've uploaded some good sequence because we came to a consensus sequence using both the forward and reverse sequences. And we got as long a sequence that we could out of that. And so that's the things that you want to make sure that you do. All right, so let's take a look at the hits from our blast. So this first window that we want to look at, this is the 100 blast hits, we'd say the top 100 blast hits of our query sequence, the sequence that we uploaded. And our sequence is right here in blue or turquoise, and it is 643 bases long. And then each of the hits is underneath it. And the top 100 are in red, and they're in red because there's enough sequence here to cover a good amount of our sequence. Okay, and so this first sequence right here, the alignment, the 643 bases of our sequence was able to align with 643 bases of this sequence. And so that is why the line is the same length. And so that tells us that the coverage of our sequence is 100%, and that would be the query coverage. But it doesn't tell us any information about how well the bases match, or about how many bases actually match, and whether or not the entire sequence matches perfectly. So we need to come down here to figure that out. So here's our top hit, and it's descriptive, so it's telling us the genus and the species. It's telling us the gene, cytochrome oxidase subunit 1, so that should sound familiar to you. It's telling you that it's a partial sequence, it's not the whole gene, and it's mitochondrial. If you look over here, the right-hand side, you see these columns, these numbers, and these are important numbers for us. And so the first number to take a look at is the E-value. The lower this number, the better. The E-value tells us the probability that this hit was based on chance. So again, the lower the number, the better that this was a true biological homology to our own sequence that we uploaded. So the value of C is great. And now if we look at query cover, I've already described what query cover means. It means that our entire 643 bases was able to be aligned with the sequence that was uploaded. So with this top hit, number one hit here, 100% of our sequence was able to be aligned. The identity is a really important number because this tells us how many of those bases are matched. Okay, so 100% identity means that 100% of our sequence matched to the hit. So the higher that number, the better. So we've got good numbers here. An e-value of 0, 100% identity, 100% query coverage. You're not always going to see that. I think one of the reasons why I got such good numbers is because I took the time to take my forward strand, my forward primer strand, and my reverse primer strand, and I developed a consensus sequence. So I developed a sequence that was as long as I possibly could get with good bases. And so we're seeing real good agreement here. And all of this is put into a score, and the higher the score, the better. Now, the longer your DNA, you may end up with a higher score, but if you look at identity and it's a low percentage, that tells you that even though you had a long piece of DNA, maybe not all of the bases were matched. And of course, the lower that percentage, the lower the likelihood that that you have a good match there. Okay, now that you know a little bit about the BLAST and how to use these numbers, now we're going to click on the top hit, and this is the alignment for the top hit. You'll see 643 bases aligned with 643 bases, and that's our query coverage. And if you looked through this, you would see that every base matches, and that's our identity. And so we got a real good match here, and we can have great confidence that this is the species of the fish that we extracted the DNA from, amplified that DNA, and sent it out for sequencing. If you take a look, this is the ascension number. That was the number that was up in this column right here. Your next step is to click on that, and you'll get a whole bunch of information about the people who submitted this sequence that was your top hit. And that'll do it for me for this video. I think you can take over from here on your own. Good luck, and we'll see you in class.